everyone, I'm Nina Brzezinski, and I'm the Grant Science Support Coordinator for NOAA Ocean Exploration, where I support Dr. Aurora Elmore and our partners at the Ocean Exploration Cooperative Institute. Thank you for joining today's NOAA Science Seminar. This is the second webinar in our four-part series about NOAA's Ocean Exploration Cooperative Institute, or OECI. Throughout the webinars, we'll meet the OECI team and hear about their exciting deep-sea exploration projects. For your awareness, this presentation is being recorded and will be posted with closed captioning on the site shortly after the webinar. You can ask questions following this presentation in the live YouTube chat box, but you must be signed into a YouTube account to do so. Otherwise, please email your questions to oeci@etal.uri.edu. Today, you'll be hearing opening remarks from Dr. Aurora Elmore, followed by presentations from Jason Fahy, Dr. Annette Govindarajan, Dr. Luke McCartin, and Val Schmidt, all falling under the very exciting theme of midwater exploration through multi-vehicle operations. You'll even get to experience some live interactions from the EV Nautilus. It is now my pleasure to introduce Dr. Aurora Elmore, the NOAA Ocean Exploration's Cooperative Institute Manager, to kick off the webinar. Over to you, Aurora.
Thanks so much, Nina, and talofa, everyone. Hello from American Samoa. It is a pleasure to speak with you all today. I'm so excited to get to tell you a bit about the Ocean Exploration Cooperative Institute, or OECI. OECI is one of NOAA's 19 cooperative institutes, which are all set up to enable close collaboration between NOAA and our external partners. I come from the Office of NOAA Ocean Exploration, which are the primary sponsors of OECI's work, and it's my role to facilitate the multitude of collaborations that we have with OECI as we work together towards NOAA's priority missions. However, OECI projects also support a wide variety of NOAA projects beyond just the Ocean Exploration Office. OECI is made up of a consortium of 5 research institutions. They're headquartered at the University of Rhode Island, which is where Jason sits, University of New Hampshire, University of Southern Mississippi, Woods Hole Oceanographic Institution, and our last partner is the nonprofit Ocean Exploration Trust, who own and operate EV Nautilus, Exploration Vessel Nautilus, which is where Jason and I and Luke are joining you from today. Oh, sorry. We'll do a wider view. This expedition that we're on right now in the waters of American Samoa has been given the name Imamana Ou Ngatafile. And this name is really important to us. It was gifted to us by the people of American Samoa, and in particular, the National Marine Sanctuary of American Samoa. The name means our waters have power, and we have seen that in the last 3 weeks when we've been out at sea. We've seen beautiful creatures, we've seen amazing new features, and we've seen some pretty powerful waves. Right now, the waters are pretty calm, so we're not being tossed around too much, but boy, these waters have been really magical. We're very lucky to be out at sea with several American Samoas who are sharing their language and their expertise with us, which has been a really great addition to our scientific plans. As you may know, we know less about the bottom of the sea than we do about the surface of Mars. And in fact, American Samoa is one of the territories of the United States and has the largest marine national sanctuary right now, and also is probably the least understood. We're very remote. We're the only sanctuary right now that is in the southern hemisphere. And it's just really exciting to have so many vehicles and so many tools brought to this unique area in order to help that group explore their oceans. In order to really understand and protect the oceans, we have to get better and faster at how we explore these important areas. To do that, we need new technologies, we need to get lots of things working at the same time, and we need to make sure that our shipboard activities aren't limited to just those of us who are on a ship, but really include people on land as well. And that is what today's seminar is all about, using multiple technologies together to increase the pace of ocean exploration. You'll be hearing from several of our uncrewed systems vehicle teams about our activities in American Samoa, and today's presentation is focused on how and why we're working hard to study the water column in the Western Pacific. So now it is my pleasure to introduce right next to me, the OECI's Associate Director, Jason Fahy, who is also with me in the control van of EV Nautilus, and he's going to explain some of the technologies we're working with before we hand off to some of the other speakers. Over to you, Jason.
Thank you, Aurora. It's so awesome to be a part of the seminar series. So thank you for NOAA for allowing the OECI to jump in. I think the first order of business is a big thank you to NOAA, not only for the invite to the seminar series, but for supporting the OECI. It's been an amazing partnership between us, truly a cooperative, collaborative relationship, and we couldn't have so many successes without the support of NOAA Ocean Exploration. Your support has been critical over the last 4 years. We've had this vision of multi-vehicle operations and how they could enhance ocean exploration. We started with our first expedition was seeing if all the vehicles could fit on this wonderful ship, the exploration vessel Nautilus. And we've grown from doing things in a series fashion to now operating vehicles collaboratively in parallel, allowing the robots to be deployed and the ship to go off to do what the ship does best, truly maximizing the value of each ship day. And it's really, really exciting to be a part of. And I think folks will hear me talk about a lot of the how we're going to do this, because the science is coming later from people who are real experts in it. But it's just super exciting how productive we've been able to be. So to introduce the technologies, I thought I'd run down a quick list, and maybe we can cut to an image of the exploration vessel Nautilus. Nautilus is an amazing platform for the work that we're trying to do. It's suited exceptionally well for mapping and ROV operations, and over the last 5 years really has come into its own as a platform to deploy all sorts of robots. And so on board with us here on this expedition, we have Mesobot from the Woods Hole Oceanographic Institution. We have the Deep Autonomous Profiler from the University of Rhode Island, and we have DriX, an autonomous surface vessel that's being launched from shore, but rendezvousing with us at all of our priority work sites from the University of New Hampshire. Super, super exciting. So maybe that's Nautilus. If we can switch to the DriX image. DriX is an autonomous surface vessel, like I said, so think a robot boat. There's no humans on board. It's controlled, for this expedition, controlled from shore. We're actually operating 100 nautical miles from American Samoa at our site here at Vailulu Seamount. And the DriX team, the operators, are sitting not only in American Samoa, but all over the country. And Val's going to tell that story, and his passion will come through for how impressive an engineering feat it is just to get this coordination going. But DriX, from my perspective, is sleek and fast. It's an amazing communications node for the vehicles that are subsea while we're operating, and allows everyone on the ship here at Nautilus to have insight to what's going on in the water column, what's going on with DriX, but also while Nautilus is off doing something else. So we can have juggling these multiple balls in the air, and we do it really seamlessly with a platform like DriX as a communications node. And then when we jump to Mesobot, Mesobot's purposely designed as this stealthy robot that operates in the midwater. If folks have been following along the last couple of years, you might see some changes in Mesobot. It got a new paint job with a little gold metal flake on the top, which we all think is awesome. But Mesobot has these exceptional cameras and all sorts of unique technologies to image the midwater. But its most impressive feature is its adaptability to carry a sensor like the eDNA sampler. And so you'll hear about the eDNA sampler from the teams that are going to speak after me. But I think in the image you can see there's a payload on the bottom of Mesobot, and I think Luke might actually show it when he talks in the show and tell. But being able to carry these payloads that allow us to get new insights into the water column are really, really important, and that's what Mesobot's great at. It's quiet. It doesn't disturb the animals in the midwater nearly as much as some other platforms, and so it's perfect for this type of work. And then because Mesobot works in the midwater, to get a perspective of the deep sea, we have the Deep Autonomous Profiler. And so the DAP is probably our least complex system from its ability to move around. We drop it off the side of the ship, it sinks right to the bottom. But what it can do while it's there is really impressive. It's got cameras and lights to image the seafloor when it lands. It's got a hydrophone to pick up the acoustics of what's going on. And we've heard whales and all kinds of interesting things. from the hydrophone. And then it's also got the ability to carry payloads, and it carries the eDNA sampler. And for this expedition, we have the first iteration of the ability to do eDNA sampling in situ deep, right? So the multi-puffer is the new device we're testing on this expedition, and we had its first deployment at our Abyssal Plain site just a few days ago and successfully sampled at 4,500 meters, which is a huge achievement for the engineering team, but also really, really valuable data for the eDNA side of the things that we do. Okay, so those are the vehicles. I think I've trying to look at my talking points here. I think I talked about the value of multi-vehicle operations, but I'll hit it again. The cost of ship time is so expensive in our business, and with limited resources and trying to get the most out of the taxpayer dollar, to support NOAA's mission, allowing these vehicles that are designed to do something specific exceptionally well, to pair them off and get the best out of them while the ship also gets the best out of. We think we are on pace to be doubling the output of a normal ocean exploration expedition. And that's just, not only are we completing the mission, but to half the cost of what we're doing is really, really something we're proud of. And so I'm excited to be out here. It's so great to be alongside Aurora. We've got a wonderful set of speakers that are going to explain to you the why we're doing it. I kind of talked about the tools and the how, ship and vehicles and up and down the water column and all that. But the why is the science part. And that's the progression that we've been on, right? We've gone from getting the vehicles to work and creating these behaviors with the vehicles to now that they can be utilized for science. And we've just been going where the science had to take us on this expedition, completely driven by the samples need to be collected. And that's like an awesome milestone. It's really operationalized this vision that NOAA and the OECI have shared for the last 4 years. So I think with that, it's my job to toss to Dr. Annette Govindarajan. Annette is in American Samoa. She's coming out on the next expedition, NA 165. She happens to be sitting at the Remote Operations Center with the DriX team. And so Annette, over to you.
Hi, thanks so much. It's really exciting to be part of this operation. Hope you guys can hear me okay. Yeah. So
yeah, all good.
So I'm happy to tell you a little bit about sort of scientifically how we're looking at what we can do with these amazing tools that we have that we've been working towards in the last several years. And so we're exploring Valulu, which is an underwater volcano in the Samoan archipelago. And it's the only hydrothermally active volcano. And just a shout out again to everybody involved before I get started. We've got the Mesobat team, which is Eric Hayden and Dana Yerger. Chris Roman leads the DAP team. Val Schmidt, who you'll hear from, is from UNH, and my biology collaborator, Santiago Herrera from Lehigh. We've got our eDNA team on the ship, Nina, Luke, and Milan. And of course, we couldn't do anything without Jason and the rest of the Nautilus and OET team. So the next slide, so we're at Valulu, which is a geological and as well as biological hotspot. Okay, and so we know that because it's hydrothermally active, chemicals are being released in plumes that feed microbial activity through a process called chemosynthesis, and that fuels biological activity, and that increases the diversity of marine life. But one question that we have is, how does this biological activity influence the biodiversity of the overlying waters? Now, we know seamounts tend to be biological hotspots. We know that upwelling, for example, of currents can bring nutrients up to surface waters and fuel biodiversity production up there. There's another process called diel vertical migration. That's where typically animals will move from the mesopelagic depths between 200 and 1,000 meters deep up to surface waters at night to feed, and then they move back down to depth to spend the daylight time. So there's these couple of different processes that could affect the exchange of biodiversity between the surface and the deep waters. So how are we going to approach these scientific questions? We're using something called environmental DNA. So environmental DNA refers to DNA in the environment. For animals, that refers to the genetic traces that they leave behind. So it's like a type of forensics approach. And so the way it works is, we go to sea and we collect our samples of these genetic traces without actually having to collect the animals themselves. We can do this through collecting water from Niskin bottles, and we can also do it with our new samplers, such as using platforms like Mesobot. Okay, and then we filter the water, we bring that back to the lab, and then we extract the DNA and we sequence that, and then we look at those DNA sequences to compare that to a library where we can put names and know what types of animals shed those sequences. So in this way, we can look at the diversity of animals without actually having to collect the specimens themselves. So the first approach that we're taking in this multi-pronged approach is with Mesobot. And so we've got, as you see Mesobot, it has a payload capacity at the bottom where we have some large volume samplers that do the filtration in situ. So that means we don't have to bring water up to the surface. They have pumps that do the filtering in situ onto large area filters. Our pumps have a high flow rate, so we can collect large volumes, larger than what we can do with Niskin bottles. And so this gives us more flexibility, which is especially important in the deep sea, where we have found that the eDNA concentrations are very low. So this allows us to collect larger samples. And we do a couple different approaches in our strategy. So we do daytime and nighttime deployments where we take vertical profiles with Mesobot from down to through the bottom of the mesopelagic up to the surface waters, and we repeat this at daytime and then do it again at nighttime, and then we can look at the difference between the two and determine who's migrating and how distribution patterns are affected by the diel vertical migration process. We also do what are called experimental dives, where are more targeted towards specific scientific goals. So I give you a glimpse of some exciting Mesobot feat earlier in the cruise, where we actually had Mesobot go into the crater of the volcano and collect samples from there. And so that watching that happen was really a thrilling moment for all of us on the ship and those of us participating from shore. It was really exciting. You can see the little yellow triangle in the middle of the crater there. So really fantastic that we were able to do that. The next approach that we're using is the Deep Autonomous Profiler, otherwise known as a lander, which can go down to the bottom. And the lander has a number of different attributes for collecting eDNA samples. It is equipped with Niskin bottles, so we can collect water from the water column, and then we bring that back up to the ship, and then that's pumped from shipboard pumps, and then we collect the eDNA that way. We also have another one of our large volume samplers, and this one is actually being deployed for the first time on this cruise. It can go all the way down. So this one can go all the way down to deeper depths, down to 6,000 meters. So down to the bottom of the sea floor, where the pressures are very, very high. And so this is really exciting that now we can take bottom samples as well as that go down to deeper depths. And so this sampler from Aquatic Labs allows us to do that. And then let me show you now a little glimpse of the data that we've collected from both Mesobot and DAP and how we've been using this to inform our sampling missions. So we have different types of sensors as well as our samplers on both these platforms. And what you're looking at on the left here is data that shows, it's called a miniature autonomous plume recorder, or MAPR, which allows us to look for the depth of the plume that's coming out of the volcano. Okay, and so we can see that around 680 meters, okay, we can see a signal that indicates a likely plume. And then we look at the turbidity data on the right-hand side collected by the DAP showing what's in the water. And we can see that below that plume depth, we see that turbidity increases, and probably representing the different particles that are coming out as a result of that plume. And then we have our third approach here, which is using DriX in combination with Mesobat. So again, one of our goals is to look at who's doing that vertical migration, okay? And we can detect layers that are migrating using acoustics, but we can't tell what species are making up that signal on the acoustics, okay? So when we look at our acoustic data, we can see that there are layers of biomass, and these are moving up and down. Okay, and if we were to sample without having knowledge of where these layers were, if we were just to pick our depths ahead of time, there's a strong likelihood we would miss the interesting layer that we want, right? So we're using this really novel, exciting approach that combines DriX with Mesobat so that we can find these layers, so that we can be certain that we're sampling in these biologically interesting depths. Okay, and so the way it works is that DriX, which is on the surface, senses with the acoustics, and we can see Mesobat, we can see the layers, and then we can participate from shore or from the ship and say, okay, this is where we want to go, and we'll let DriX know, and then DriX will talk to Mesobat. Mesobat will go do what it's told, and we can see that from DriX, so it's verified. And so we've got basically cooperation between different vehicles as well as people based on the ship, and as well as long distance. So this is the lower image shows where I was participating in this from Woods Hole when we were doing this off of Hawaii on our last year's cruise. So it was pretty exciting. So we collect our eDNA samples with these various approaches to allow us to look further at our, the question of how the volcanic activity is influencing the biodiversity in the water column. We get the samples, but that's actually when our work just begins, because we have to then take the samples and process them. And the first step in processing them is on the ship, where we take those filters. And actually, if you look on the right-hand side of the slide, you'll see a filter that's really discolored from all of the iron oxides as a result of the plume. Okay, and then Luke McCartin will tell you more about the process that they're doing on the ship in terms of handling these samples. And then ultimately, we're going to ship them back home to our labs and do some processing there. But now I think I will hand this over to Luke to tell you about these next steps.
Question that I think some of our viewers would be really interested in. You mentioned at the beginning that you were at Woods Hole out on Cape Cod as we were starting the expedition here almost 3 weeks ago, and then
Right.
you made your way to Pongo Pongo because you're going on the next cruise. Could you tell us a little bit about what's happening on NA 165, which is the next cruise in American Samoa after this one?
Yes. So it's really exciting because we're going to have use our new eDNA sampler, the one that's currently on the DAP that can go to the deeper depths, and we're going to put that on an ROV called Hercules, and that'll be the first time that we do this, and it'll allow us to collect even more eDNA samples, and we can use the information that we've collected on 164, this current cruise, to help us guide what and where we want to sample in those strategies. So this is really just an amazing opportunity to have so much knowledge about this really exciting scientific system, and I can't wait to go on the ship, and I can't wait to see what we get when we come home in our samples.
Thanks, Annette. Yeah, it's really amazing to even see what we got a few days ago, really, that you're able to show here. And
Yeah.
I think also having scientists who are onshore enables us to have a lot more people involved in this kind of expedition, but also really helps us do 24-hour operations, because we only have so many people on the ship, and folks need to sleep every once in a while. We let people sleep. So having you in Santiago chiming in at all hours has been really, really helpful for our success on this expedition.
It's been amazing how connected we feel with the team. Both here now I'm physically here in Pago Pago, but also the team on the ship, and we use our WhatsApp, and we are just, texts are flying 24 hours a day, and it does really make us feel connected, and as part of the whole team.
Yeah, and we talk a lot about the uncrewed systems vehicles as some of our tools to conduct ocean exploration, but boy, we have such an amazing tool in all of the scientists and all of the people who are working together really collaboratively to make these exciting things happen. So thanks so much, Annette. Looking forward to seeing you in a few days. While Annette's been on shore, one of her colleagues, Dr. Luke McCartin, who was a guest student in Annette's lab during his PhD, is now a postdoc at Lehigh University, and he's been out with us on Nautilus for the last few weeks and is currently now in the wet lab. So we're going to turn over to Luke. There he is, down in a very quiet looking wet lab at the moment. We're used to seeing it just crowded with lots of people. So Luke, tell us what you're up to.
Yeah, thank you so much, Aurora, for the introduction. Everyone went to bed at about 4 a.m., so they're resting. We've had a very busy expedition. Like Aurora said, like you've heard, so I'm joining you from the wet lab of the exploration vessel Nautilus, where we've been collecting over 300 samples for eDNA analysis here on this expedition in American Samoa. And so you heard from our expedition leader, Jason, and from Dr. Annette Govindarajan about how we're using multiple platforms on this expedition to collect samples. So I thought I'd kind of show you a little bit up close at actually how these samples get collected. Before I do that, to give you a little more scientific context, like Annette mentioned, we are studying organisms in the ocean twilight zone, and particularly we're studying them in the waters of American Samoa over abyssal plains, off the shores of islands, and then also over this seamount by Lulu'u. And now I just want to say that I know we got weathered out a little bit, so we had to work in the lee of some of the islands, which wasn't our original plan, but we had some pretty awesome views over the course of this cruise, so it's really been a joy to work in these waters. And so I want to just spend another second to talk a little bit more about Lulu'u. So Lulu'u is a super interesting system. It is both a seamount and an active volcano. During my PhD in Dr. Santiago Ferreira's lab at Lehigh University, I actually was involved in a study that he led where we showed that the eruption cycle on the seafloor here can actually serve as a natural laboratory to study benthic organisms and how they colonize the deep seafloor. And like you've heard, it also has active hydrothermal vents. But here on this expedition, again, we're studying this system from the water column. So we're looking in the water and seeing how the organisms that live in the ocean are interacting with this volcano. And so to do this, we're sampling in a non-invasive way. So we're not actually taking any physical collections of organisms on this expedition. And so you've heard about the two instruments we're using for eDNA sampling, the Deep Autonomous Profiler and then the Mesobot. And so with the Deep Autonomous Profiler, or the DAP as we like to call it, we take samples with Niskin bottles, and then we take that water and we filter it on the deck of the ship. In addition to filtering that water for environmental DNA analysis, we're also conducting some sampling to take chemical measurements, like the concentrations of trace metals. And so having those data give us more of a perspective on the broader biogeochemical context in which we are working and studying life in the ocean. So I like to think that we've really perfected the art of sampling eDNA on the deck of the ship. I first started doing this about five years ago on a cruise with Annette's lab and also with my advisor Santiago's lab, where we worked with the Mesobot, but also with a CTD rosette. And so even though we've really had a lot of practice doing this, sampling water from Niskin bottles on the deck of the ship takes a lot of time. So to give you an idea, I think our record is about 3.5 hours to process 18 Niskin bottles on this cruise. But what this sampler that you're seeing here next to me allows us to do is not only sample much quicker, but also sample with less interaction from the user, so us, and then also, well, us physically, so we interact with it remotely, the ship, but we don't actually touch the samples as they're being processed. And it also allows us to take samples a lot quicker. So to process these samples off of this sampler, you know, our record time on this cruise is about just an hour to get through around 16 samples. So it's a lot faster. And so some of the advantages of using this system, and this specific sampler is called the WHOI Oceanics multi-sampler, is it allows us not only to take more samples and to process them more quickly with less interaction from us physically. And that last part is important because the more interaction we have with the sample, the more possibilities there are for us to potentially contaminate the sample. So it not only allows us to do that more quickly, but also we can collect more samples. So each of these samplers holds 16 different filter capsules, and we can actually daisy chain them on Mesobot so that we can collect 32 samples over the course of a single AUV dive. Now, when we're collecting these samples, not only are we collecting more, but also in larger volumes. So like Annette said, these samples collect very large volumes of eDNA. Over the course of just 20 minutes, we can actually filter 50 liters of seawater. So if you do the math there, over the course of a deployment, we're potentially collecting over 1,500 liters of seawater. And some of the work that I was involved in during my PhD that was led by Annette actually demonstrated that taking these large volume samples gives us a much more comprehensive view of biodiversity in the water column than taking a smaller volume sample with Niskin bottles. And so in addition to this sampler, like you heard, we also are using another sampler called the multi-puffer from Aquatic Labs, which can go down to 4,500 meters deep. But really, our work begins in the wet lab once the vehicle gets on deck. And so normally, this is attached to Mesobot, and we'll actually go out to Mesobot and take it off and to take our samples. But I brought it in here to give you a closer look, give you a bit of a demonstration. So the first thing you'll notice is that this device here, this is actually the filter capsule. And so we're always gloving up so that we don't potentially contaminate our samples. What we do is we go out to Mesobot and we pull these filters off. And so hopefully you can see this here. And basically how this works is it interacts with the sampler through this housing here with an O-ring so that it seals. And then it has a one-way valve on the front. And so this one-way valve makes sure that water doesn't actually enter the sample until we want it to, or until we're actively pumping water. And so once we get these off, we will clip the zip tie, because zip ties are used for everything in deep sea research, and we'll pull them off to get it out of the housing. And then we actually devised this method of reducing the volume of the cartridge here so that we can preserve it for shipment back to our lab. So I fortuitously found this pipe cutter, or this model of pipe cutter, on the Atlantis in the forward hold one day when I was looking to try and open one of these things up. We decided that it was the perfect fit for our filter capsule, so I got really lucky on that cruise, and now we've purchased them. And yeah, we talked about calling this like the eDNA Cracker 2000 or something like that, because it's becoming part of our normal laboratory protocols. And I'm very proud to say, and I'll show you quickly how this works, that we've trained a bunch of people on the ship to actually open these filters, which is fun, so that I don't have to sit here cracking filters by myself the whole time. It's not too bad. It's a little squeaky, if you can hear that, because we've gone through a lot of samples. We need to add a little WD-40. But what happens is that we actually are able to open this filter capsule here, right? And then we pull out this filter. And so this filter membrane, which hopefully you can see, how it works is it's kind of folded like an accordion around the filter capsule. And so any particles that are captured on this filter can be extracted back in the lab. And so essentially 50 liters of seawater, which is a large volume, right, is concentrated over this filter. And so the picture that you should be seeing now is not eDNA, right, because that's pretty tough to see, although we can certainly see some color from chlorophyll when we take samples. But these are iron oxide particulates. And so this just demonstrates the concept. So these filters have allowed us to concentrate iron oxides over it, and you can see that we can get all of these, all this material so that we can extract it back in the lab for environmental DNA analysis. And so this has been a lot of work, right, over the course of this cruise, processing 300 samples and cracking them open and then freezing them for shipment back to the East Coast has certainly taken a lot of work on our end. But I'm just so fortunate that we had such a fantastic team working with us. So we also had another postdoctoral researcher, Dr. Nina Yang from the Woodsville Oceanographic Institution, Malong Sengtep, who's a graduate student at Lehigh University, where I work, Moranke Harris, our science manager, and then Leo Eleato, who's the ocean science intern on this expedition, and Talofa Fahy and Ella Ashford, who are working on behalf of the National Marine Sanctuary of American Samoa. And so coming out to sea and knowing you're about to process a few hundred samples can be pretty daunting, but I want to say that it's really been fantastic to work with everybody. we've set some record times for sure, and we've processed a lot of samples, and I'm incredibly fortunate not only to have the chance to work with these folks, but also to learn a lot from them in the process. And so, yeah, once we get these samples back to the lab, we will be analyzing them to see what organisms live in the water column. We'll go all the way from microbes, so those microorganisms that will actually capture whole cells on the filters, all the way up to traces of DNA that get captured as well. And so with that information, we can start to assess the biodiversity in the waters of American Samoa, including here in the National Marine Sanctuary at the Wailulu Seamount. And so thank you so much for tuning into the Wet Lab. And with that, I will turn it back to Jason.
Luke, nice job. Thank you. Not only was it scientifically interesting presentation from Luke, but he nailed the demonstration. These are always fraught with risk, and he was handling the pipe cutter and doing all that. Just, I mean, he's done it hundreds of times, but it was just really impressive. So kudos, Luke, and to the whole team. Great job. I will say we've encountered some rough weather on this expedition, and the most, I think, maybe recovering the deep autonomous profiler and then the sample handling is the most sensitive to sea states. And so the team in the wet lab has been doing what is very delicate work, working with very sharp tools to cut and extract these filters under really challenging conditions with the ship moving around. And they've done it exceptionally well. Just a total team of professionals. Even the interns now are like holding court in the lab about how we do things. You know, like I'm not allowed in there basically because they're so good. So that's been amazing. We heard from Annette about the eDNA and the biodiversity studies that we're doing. We heard from Luke how we're handling the filters. One of the new interesting innovations that we have devised over those last 4 years of working with these vehicles together is this verified directed sampling. And the key enabler there has been the DriX, this autonomous surface vessel. So to tell us a bit more about DriX, we're going to toss it to Val, who's back on shore in Pongo Pongo. And Val's going to tell us about this amazing autonomous surface vessel. Over to you, Val.
Hi, everybody. Welcome to the Remote Operating Center in Pongo, American Samoa. We call it Rock 1. Let me start my presentation by introducing you to DriX 8. So DriX 8 is a 8-meter long vehicle, maybe about 3/4 of a meter wide, and it looks like a submarine, and maybe it should go underwater, but it doesn't. It stays on the surface, but it is super hydrodynamic and very efficient, and a long endurance vehicle for its size. You can see all those antennas on the top. Those are for communicating with the DriX through various modes of operation and navigation systems for knowing exactly where DriX is. There's a marine radar on the front, that white dome that allows us to see through fog and haze and ships far in the distance. You can see the Starlink antenna on the stern that gives us a satellite link to literally connect from DriX anywhere in the world. And then the part that you can't see is a keel down below the water and a big gondola. And that gondola holds sort of the main science sensors that come in the form of a USBL system for tracking underwater vehicles and communicating with them. And then an EM712, a seafloor mapping sonar, and an EK80 water column mapping sonar. Let me give you a sense of some of the data from those. This is data from our EM712 that was taken last year over McCall Seamount. It's a really capable system and the very first of its kind devised, engineered for a small ASV like this, with penetrations down below 2,000 meters into the water depth. And then the primary sensor for this mission is the EK80, which is a midwater mapping sonar used commonly for fisheries, but picks up little critters in the water column and allows us to give context to mesobot sampling efforts and the analysis that Annette does, and allows us to direct mesobot through this sort of cloud of stuff in the water column to sample in maybe a layer or outside a layer, and really provide meaning to the measurements that they take. But let me say, we usually operate DriX from sea. Everything that we've done to date has been off of the Nautilus, and we gave ourselves a new challenge to expand our scope of operations on this go, and that is to operate DriX not from sea, but from shore, over the horizon, via satellite, with operators spread out all over the place. So this is Rock 1 here in Ponga Ponga with KG Fairbarn, who's one of our engineers and DriX pilots. And then we have another operator station to provide some redundancy to this one back in Durham, New Hampshire, on the other side of the globe. This is the team there who have a similar kind of setup at the Center for Coastal and Ocean Mapping at the University of New Hampshire, and they're maintaining a watch, one person there and one person here, 24 hours a day, every moment that the DriX is deployed. You might know that it's winter in the southern hemisphere, and it's not cold here, don't get me wrong, but we're definitely feeling the seas from the far south, and that they've exceeded 3 meters significant wave height a lot of this mission. And when you're operating DriX from sea, or from shore, well, at any time, that's a real challenge. In fact, the seas have been so high, if we'd have been operating from the ship, we'd have never been able to deploy the DriX, except for maybe today. It's pretty calm out there. But just to give you a sense of what the swell conditions are like and the wave conditions, the winds have exceeded 25 knots on a lot of our first goes doing this, and it's been nail biting at times. If this image doesn't convince you that it was rough out there, the next one might. It's a shot off of an iPhone, but you can see the horizon there. I apologize for the antenna of the radio sticking up in the middle of it, but the seas in the Southern Ocean are no joke. Let's see what else. So we're currently on a multi-day mission, and I want to give you a sense of what that looks like. So we've been out for more than 24 hours, no, more than 48 hours, and I'm going to sort of zoom into the operator station and give you a sense of what we've been doing. So in the upper left, you can see an image of DriX. This is from one of our first missions in the lee of Tutuila, and Nautilus is there with us in the map in the upper left. And then in the center bottom display is the EK80, and it's going, and we're tracking Mesobot and going through the motions of making all of this work and sorting out all the comms and providing all of that context. Okay, so this is not our main study site, of course, here in the lee of the island. Our main study site is Vaiuluu, which is about 100 nautical miles offshore. And when you're operating 100 nautical miles offshore, you got to think really carefully about everything that can go wrong. And that's really been the focus of this research effort from an operations standpoint, is how do we do that? How do we assess all the things that can go wrong, have backups for the backups, have spares for everything, and make it all work? And it has taken a team of folks from all over the world contributing to this at various times. I guess I have one more view of, so this is a view of a remote operating center, and part of the team working through issues while the DriX is at sea. The likelihood of failure when you're in 3-meter seas is way more than it is when it's flat calm. And we've honestly never operated DriX in conditions like this, and trying to assess what those failures are going to be and the impact, and then mitigate that has been a process that started more than a year ago, working with other operators of DriX vehicles and shoring up all of the bits and pieces. But while we've been here, we've been blessed to have great support from University of New Hampshire back at home, and then in France. And if you can believe it, even our director of our lab has chimed in and to help out. And we have an image that he sent just last night of him on an icebreaker in the Arctic off of Greenland, tuning into DriX operations and watching from from there. So it really has been a team effort from all over the world, bringing it all together. And sort of the final go, I'm just going to show you kind of a nice video of DriX floating here in the harbor. It's kind of beautiful, but it was a late night go for us as we were testing systems to make sure that if our Starlink, our primary sense of communications, fails, we have backup systems in place. We've spent a lot of time making sure that all of that works. We can start the engine if we have to. We can turn things on and off if we have to fix things. We're trying to make sure that if anything goes wrong, we have a way to find DriX. We have a way to get it back home safely. So I think that's all I've got. And with that, I'll hand it back to you, Jason.
Thank you. I know the conditions have been challenging, but your point about if DriX was aboard Nautilus, as we've operated for the last 3 years, likely wouldn't have been able to deploy. And now with being able to operate from safe harbor and leave, and how resilient DriX is to sea state, a lot of this is credit to you and your team of how prepared y'all are to do this. But it's just been really, really exciting.
Honestly, we couldn't do it without the Nautilus, and we wouldn't do it without the Nautilus. Nautilus is our backup out there 100 nautical miles offshore, and although we can raise the weather window when we operate from shore, our limit is always going to be what do we have to do to get DriX back if something goes awry, and that's a credit to the ship and the fantastic deck team that they have and their ability to operate in those kinds of conditions. So we've been really thankful to have all of that support.
Val, we got you. Don't worry. So I guess maybe a real-time update, right? Currently we have DriX on station at Vailulu. We have Mesobot about to be deployed for another verified directed sampling kind of daytime mission. This will be the last one of the expedition, and then DriX will transit back, and we'll probably be following right behind as we head back into port at the end of NA 164, and then we'll do a bunch of fun shoreside events. Yeah, we will see shoreside events, and then NA 165 comes on, and they get to take the ROV down into the crater. And so, folks, tune into Nautilus Live to see from inside Vailulu Seamount live over the following month, the month of September. I think now we're headed into the question and answer portion of our program. The team on shore supporting us has been collecting questions through the email address or through YouTube if you've logged into the chat in YouTube. Here's my first question. Holly asks, what happens if a robot breaks while others in the water working? This is precisely the scenario that Val was hinting at. We've talked deliberately, maybe Val can chime in here too about some of the nuances, but we had a number of meetings. We walked this through on the deck numerous times, talking about how we prioritize and how we handle the recovering of a vehicle that might be in trouble, or recall the vehicles that are in the water to go support the other one. And it is a topic that we don't take lightly. Val, you've led a lot of those conversations. Do you want to maybe add a little bit?
Yeah, like we've done like role playing where we envision that something goes wrong, and then and we sit down as a group and talk through like what the sequence of events might be to try to prepare for that very thing. And you're right, like we sit with the various vehicle operators and talk about their strengths and the weaknesses, and how much time they could be in the water unattended if we had to attend to another, and working with the ship to try to prioritize those things. So it's a constant conversation, and one that we revisit as we do different kinds of operations in different places and those situations change.
Val, maybe you could also tell the story of the failure that we had early on in the expedition with the Starlink update. And I don't say that with any sort of malice, but we talked about all the different contingencies, and then here you were confronted with one that seemed benign, but yet snowballed into just a really, really complex problem to solve.
Yeah, it turns out that marine versions of Starlink will update their firmware. They tell you that it's going to happen in advance, but there's nothing you can do to stop it or delay it. And we noticed that the Starlink on the DriX had been queued up for a firmware update, and so we knew it would come. There was nothing we could do about it, so we applied the update. And when we did, the Starlink did not come back operational, and it was a nail-biting moment that was plagued by other issues that we had not seen before, that only would have occurred if we were at sea and Starlink had failed. And so we learned the hard way that there were some things that we thought were working that were not, and we limped our way from about halfway, maybe 2/3 of the way between Tutuila and Manua Islands to an island called Ofu. We got a 4G signal there because they have cell service on the island, and and then got into the lee of the island, and Starlink eventually came back. But in the forensic analysis, which we spent a lot of on the last few days trying to understand exactly what happened, we sorted out that the movement of Starlink when it first boots up, it makes it difficult for it to connect to the satellites, and the firmware process never fully completed, and the dish flipped back and forth between two firmware states probably 17 times over the course of like 8 hours, and we spent a day in the harbor when we got back testing all of the backup mechanisms that allow us to pilot the vehicle when we don't have Starlink, and all of the other things that can go wrong, and the situational awareness for all the operators. So it's one of those problems that is a real challenge, and one that we need to think carefully about, and also exercises like forces us to actually, well, it just reveals shortcomings and things that we think that are working and that we had tested in the past, but in a very particular circumstance, have an edge case that's hard to fix. So learning lesson for sure.
And Val, Nautilus had turned around. We were on our way back to do a rescue, and then DriX and your very capable and late working team were able to get things back online thanks to that connection at a nearby island. It really was exactly what we'd planned for in that sense. We knew if you were in trouble, we were going to come to your aid, and we were at Vaililu, and we headed back towards you and actually didn't make it to you when you'd already done the fix. So then we came back out. So it was
that's right.
a fun
And
little jaunt for us.
ironically, like we envisioned this particular scenario, and we provided a remote controller for the ship and trained the deck team on how to pilot the DriX around, because in this one type of condition, they could have piloted the DriX back to shore without the Starlink connection. And so we were totally thankful you were out there and you were willing to come and help and bail us out. And then also kind of proud that we were able to fix it and we could send you back to the mission, and we could go home on our own.
Yeah. We have another question that's come in. How many scientists, engineers, etc., are required to operate one of these vehicles? And I guess we'll hand that first to Val, and then I'll hand it over to Jason to answer as well. Val, how many people do you need? They're all over the world, but how many is it?
They are. So part of it is watch standing. So we have 2 people on watch at any one time. We're running a 3-section watch in Durham in New Hampshire. So that's 3 people. And then a 4-section watch here. So that's sort of 7 for round the clock, 24/7 operations. We have a graduate student here, and she's helping out with all kinds of stuff and sitting under instruction watch. And then we are thankful to have support from folks in France who are not really part of the operation, but are part of the manufacturing team of the vehicle, and they've been helping out from time to time to provide support. So it seems like a lot of folks, but for a 24/7 operation on the far side of the planet, I think we're doing pretty well.
So for the other vehicle teams, overall on Nautilus, we have 27 people in the science party. That covers everything from all the ship science equipment that necessarily wasn't the primary focus of this expedition, but then the eDNA sampling team. The Mesobot team is 3 people, hands on the vehicle, and then the Deep Autonomous Profiler has a 2-person team on board. And so it takes plenty of humans to operate the robots. And I think DriX is a perfect example of how we can move a lot of those human components to shore for certain functions, but then also in this problem scenario that DriX had to come back in, it's going to take people hands-on to be able to work with the vehicle, turn wrenches, swap out components, and we're still, part of this research is finding that perfect balance between those 2 things. Who can be remote, how many do you need on shore? Because in a situation like we are here in American Samoa, where if we had open bursts, we could have more folks from the local community. We could just tell the story of what we're finding that much clearer and enriching experience by having those people on board. We're really looking forward to that. And so that's kind of a secondary driver for everything we're doing with these multi-vehicle operations. More questions? Yeah? Okay. It looks like we've got a few more minutes. From Sherry Lynn, about how many animals or organisms have been sequenced, i.e. how big is the database for eDNA? Great question. I think if Annette is still at Rock 1 with Val, maybe Annette would be great to answer this one.
Hi. Yes, that is a great question, because first, to just explain why that's important for those who don't know, when we get an eDNA sequence, it's just the sequence A's, T's, C's, and G's. We don't know what it is. We need to have a database where animals that have already been identified accurately have been sequenced, so we can compare our eDNA to that reference database so that we can put a name on it. So it's really important that we have the database. The database is incomplete in general. It's in here as well as in most areas, and we need to have parallel efforts going on to build up that database. And we've been doing some of that through WHOI. We've putting a big effort through WHOI's complementary Ocean Twilight Zone program, where I've been focusing on fish, where we've been getting fish specimens from the deep sea and then getting them identified and doing getting the barcode sequences from that. So it's a work in progress. It's important that that is done in parallel. Oh, and we see the great video from now with the shark. And actually, so one of the really cool things, this is kind of tangential to the question, but it's really the cool thing about eDNA, is that we can see animals like sharks as well as microbes from the same sample stream. And so that's kind of what makes one thing that makes eDNA analyses really unique, is that you can look at, all of the domains of life from bacteria to sharks and to these, cool jellies, all of them from one sample, right? And that's something, you can't do, say, with nets, right? You might get some of the zooplankton, but you're not going to get the shark, and you're not going to be sampling the microbes. And so that's something really cool about the eDNA. So tacking that interesting fact to the answer about the very good question about the reference databases. So Aurora, Jason?
Yeah, and Annette, maybe if I follow-on question, can Mesobot help with the building of the library by, seeing animals on video and then you having an eDNA sample? Does that help you make the connection to what that mysterious eDNA sample might be, or does it take having, like you said, a deep sea fish sample on the lab deck or on the lab table and then taking samples that way?
A voucher.
Yeah, we still need to have the reference database. The video can be very helpful, but a lot of times you need to, to do the species level identifications, you really need to have the morphological specimens and a taxonomist who's trained in looking at the morphology. But having the lights on allows us to compare different approaches and allows us to, expand upon our overall knowledge of the biodiversity. So they're very complementary.
The sharks out here have been just as curious about our robots as we've been about them. I think we're doing a little science exchange with the biology here at Vailulu.
Yeah. One thing we do with the lights, just to say that we've been doing on this cruise, is we keep the lights on, and we think of the light. A lot of animals are attracted to the light, and so one thing we've tried in some of our experimental dives is keeping the lights on, seeing if we can attract more creatures, and if then we can pick up that signal in the eDNA, right? And that might allow us to, sort of more fully explore and see what we might be missing out there. So it's really, really cool.
Thanks, Annette. Yeah, that was 3 different videos from Mesobot that have been shot on this expedition. There was an oceanic white tip, followed by some pilot fish and a bunch of salps and tiny little plankton swimming around in the black and white one. It's been really awesome to see all the biology firsthand.
I think we're at time. I'll close by just a huge thank you to all the participants, not only on board, but the speakers in today's seminar series. If folks are excited by what they heard in today's seminar, tune into Nautilus Live. Like literally when we get off the seminar, tune in, because we'll be launching Mesobot. DriX will be on station. You'll hear through the audio channels the conversations between the operators about selecting what they're going to do and the layers they're going to, you'll see verified directly sampling kind of happening live right after this. And if that isn't interesting enough, NA165 is coming up at the beginning of September. Again, yeah, nautiluslive.org, follow along ROV expeditions. I'll be home, but tuned in, like glued to the screen. And so I'm really excited to see how well NA165 goes. Nina, I think this might be back to you to wrap it up.
Yeah, thanks, Jason, and everyone for those great presentations. I just want to say we're really fortunate to be able to have live interactions with the Nautilus today and speak with you all on shore as well. So to close out, I do want to thank you all for joining us to learn more about the Ocean Exploration Cooperative Institute. And I'd also really like to thank our OECI partners and NOAA collaborators who make all this work possible, especially the University of Rhode Island Interspace Center who produced the webinar today. And as I mentioned earlier, this webinar is the second of a 4-part series that's highlighting the amazing work of OECI, and details on the upcoming webinars can be found using the website that will hopefully pop up on your screen. There it is. Thank you. And that being said, I hope you'll join us on September 20th at 2 p.m. Eastern Time for our next OECI webinar that's entitled Education and Outreach, Building the Blue Economy. Thank you for joining us today, and we look forward to seeing you next time.